Welcome back to BiochemSerye Learning on the Go.
Learning on the go.
This is podcast 2E, Isomerism in Carbohydrates.
Oh, isomerism.
Yeah, as a biochemistry professor, I've seen countless students struggle with this concept. But it's truly foundational to biochemistry.
Absolutely, I agree.
It's like trying to understand a language without knowing the alphabet.
Yeah, that's a great analogy. And it's especially critical in carbohydrate chemistry.
Right.
We're talking about molecules that fuel our bodies and store genetic information.
Right.
And even provide structural support. And isomers are at the heart of understanding their diverse roles.
Exactly. But before we dive into all the different types of isomerism, maybe we should start with just a quick recap of what isomerism actually means.
Sure.
Just to make sure everyone's on the same page.
Absolutely. It's all about arrangement. So imagine you have a set of building blocks, in this case, atoms.
Yeah.
You can use those same blocks to create a whole range of different structures.
Okay.
And that's what isomerism is. Molecules with the same chemical formula but different structural arrangements leading to different properties.
Okay, so it's like having the same ingredients but making completely different dishes.
Exactly.
You might have flour, sugar, and eggs.
Yeah.
But you can use them to bake a cake or make pasta.
Precisely. And just like a cake and pasta have different textures and tastes,
Right.
isomers can have vastly different chemical behaviors and biological functions.
Okay, I'm with you so far. So let's get into the types of isomerism we see in carbohydrates.
Okay.
First on the list, we've got Ketose-Aldose Isomerism.
Right.
What's the key distinction here?
Here we're focusing on the carbonyl group, which is a carbon double bonded to an oxygen.
Okay.
In aldose sugars, the carbonyl group is at the end of the carbon chain, forming an aldehyde.
Okay.
Think of glucose, the sugar our cells use for energy.
Got it. Aldehyde at the end. And what about ketoses?
Ketoses have that carbonyl group within the carbon chain, forming a ketone.
Okay.
A prime example is fructose, the sugar found in fruit.
Ah, fructose. Now, I remember from teaching that even though glucose and fructose share the same formula, C6H12O6,
Right.
they have distinct properties, right? Fructose is much sweeter, and our bodies process it differently.
Exactly. That difference in sweetness and how our bodies metabolize them stems from that seemingly small structural variation of the carbonyl group. It highlights how a single change in arrangement can have a big impact.
Fascinating. Okay, let's move on to another type of isomerism, D and L isomerism.
Okay.
This one always trips students up because it involves visualizing molecules in 3D space.
It definitely requires some spatial thinking. Think about holding up your hands.
Okay.
They're mirror images, right?
Right.
But no matter how you twist them, you can't superimpose one perfectly over the other.
Right. They're like those trick mirrors that flip your reflection.
Exactly. That's the essence of chirality. It's handedness that some molecules have.
Right.
And D and L isomers are like those mirror image hands.
Okay.
They have the same atoms connected in the same order, but their spatial arrangements are flipped.
So how do we tell a D sugar from an L sugar? I know it has something to do with a specific carbon atom.
You're right. We look at the chiral carbon furthest from the carbonyl group.
Okay.
It's all about the orientation of the hydroxyl group, the HOH attached to that carbon. If it's on the right, we have a D sugar. If it's on the left, it's an L sugar.
Okay, so picture that chiral carbon. If the hydroxyl group is on the right, it's like giving a thumbs up. That's your D sugar.
You got it. And for our listeners, if you need a visual, try holding up your hands with your thumbs pointing out.
Okay.
One hand represents the D isomer and the other represents the L isomer.
Great tip. Now, I've always been curious about why most naturally occurring sugars are D isomers.
Yeah.
Is there a reason for this preference?
It's a fascinating example of biological selectivity.
Okay.
Enzymes, the catalysts that drive biochemical reactions, have evolved to interact specifically with D sugars.
Okay.
It's like they have a lock-and key mechanism that only works with a specific handedness.
So those L sugars are just left out of the party.
Essentially. They simply don't fit the active sites of most enzymes involved in carbohydrate metabolism. It's a beautiful example of how evolution has shaped the molecular world to favor certain structures over others.
Okay, so we've covered ketose-aldose isomerism and D and L isomerism. What's next on our isomerism tour?
Let's talk about a phenomenon that demonstrates the 3D nature of these molecules. Optical isomerism. Remember how we said chiral molecules have this handedness?
Yes.
Well, that handedness affects how they interact with light.
Wait, they can actually twist light?
Yes.
I'm picturing a sugar molecule with a tiny lightsaber.
They'll love the image. It's not quite a lightsaber,
Okay.
but you're on the right track. These chiral molecules can rotate plane polarized light.
Wow.
Some isomers, called dextrorotatory or plus, rotate light to the right.
Okay.
Others, called lavorotatory or twist it to the left.
So it's like each isomer has its own light-bending superpower determined by its specific 3D shape.
Yeah.
That's how scientists can differentiate between them, right?
Precisely. By measuring how much the light is rotated and in which direction, we can identify and distinguish between different isomers. It's a fundamental technique in chemistry and biochemistry.
Now that we've touched upon how these isomers interact with light, let's move on to epimerism. It sounds a bit intimidating, but isn't it just a minor variation on a theme?
You could say that. Epimers are diastereomers, meaning they're not mirror images of each other. But the key here is that they differ in configuration at only one specific chiral carbon.
So they're like two recipes that are almost identical,
Yeah.
except one calls for a teaspoon of cinnamon while the other uses a teaspoon of nutmeg.
That's a delicious analogy.
Uh-huh.
And just like that small change in spice can alter the flavor profile, that single variation in configuration can significantly impact the biological behavior of epimers.
Do you have a good example from the world of carbohydrates?
Absolutely. Let's consider glucose, galactose, and mannose. They're all 6-carbon sugars and epimers of each other. Glucose and galactose differ only at the 4th carbon atom, while glucose and mannose differ at the 2nd. It's just that one tiny difference, but it matters.
So it's like they're playing a game of molecular musical chairs, and only one hydroxyl group is switching seats. But even that seemingly minor change can have a big impact on how these sugars interact with enzymes, right?
Exactly. Enzymes are incredibly specific. They've evolved to recognize and bind to particular isomers, and even a slight change in configuration can disrupt that interaction.
This is incredible. It's like opening up a whole new dimension of carbohydrate chemistry. But we've got one more type of isomerism to cover. Enantiomerism. This one always brings to mind those beautiful ring structures.
Yes, enantiomerism is all about rings. You see, while we often draw sugars as linear chains, in reality, many of them prefer to exist in cyclic or ring forms.
So they're ditching the linear lifestyle for a more compact, circular vibe.
Exactly. And when a sugar molecule forms a ring, a new chiral center emerges, which we call the anomeric carbon. It's the carbon that was part of the carbonyl group in the open chain form.
Right.
Now it has 4 different groups attached to it, making it chiral.
So cyclization introduces a whole new level of chirality to the mix.
Yes.
This is getting complex.
It is, but it's also incredibly elegant. Now, because of this new chiral center, we get 2 possible configurations, alpha and beta ionomers.
Okay, I'm visualizing a ring now. Where do these alpha and beta anomers come into play? It has something to do with that hydroxyl group on the anomeric carbon, right?
You're right on track. In an alpha anomer, the hydroxyl group on the anomeric carbon points downwards, below the plane of the ring. In a beta anomer, it points upwards, above the plane of the ring.
So it's like the hydroxyl group is deciding whether to hang low or reach for the sky.
I love it. And just like with other types of isomerism, this seemingly small difference between alpha and beta anomers can have a big impact on a sugar's properties and biological activity. For example, the difference between starch and cellulose, both polymers of glucose, comes down to the anomeric configuration of their glucose units.
Okay, my brain is officially full.
Aha.
We've covered a lot of ground from Ketose-Aldose Isomerism to the complexities of enantiomerism.
We have, but it's all connected. Understanding these 5 types of isomerism is essential for grasping the diverse roles that carbohydrates play in biological systems.
Before we move on to some real-world examples, let's quickly recap the 5 types of isomerism we've discussed so far.
Sure. We started with ketose-aldose isomerism, where the position of the carbonyl group differentiates aldoses and ketoses. Then we explored D and L isomerism, which deals with the handedness of molecules and the arrangement of atoms around a chiral center. We delved into optical isomerism, which explains how these chiral molecules can rotate plane polarized light. Then we tackled epimerism, where isomers differ at only one specific chiral carbon. And finally, we unraveled enantiomerism, which arises from the cyclization of sugars and the formation of alpha and beta configurations.
That's a great summary. Now I'm ready to see how all this theoretical knowledge plays out in the real world. I bet you have some fascinating examples lined up for us.
Oh, I do. Get ready to be amazed by how these subtle structural differences have profound consequences for everything from how our bodies metabolize sugar to the structural integrity of plants.
Can't wait.
All right, are you ready to see isomerism in action?
I am. Let's do it.
Okay, let's start with something we all know and love, sugar.
Oh, sugar. All right.
Specifically, glucose and fructose. We've already touched on them as examples of ketose-aldose isomers.
Yes, glucose being the aldehyde
Right.
and fructose the ketone, despite having the same chemical formula.
Exactly.
But how does that play out in our bodies?
Yeah,
We eat both, right?
exactly. And that's where their different structures lead to fascinating differences in how they're processed.
Okay.
Glucose, your classic aldose, is the primary fuel for our cells.
Right.
It travels in the bloodstream, it gets taken up by cells, and is broken down to release energy. Think of it as the go-to energy source for most of our tissues.
Okay, so glucose is like the all-purpose fuel that keeps our engines running.
A perfect analogy. Now, fructose, the ketone sugar, is a bit different.
Okay.
While we can metabolize it, the primary site for fructose metabolism is the liver.
Okay.
It's used to replenish liver glycogen stores and can be converted into glucose or fatty acids.
Interesting. So even though they're isomers, their metabolic feats are quite distinct.
Right.
It's like they have different job descriptions in the body.
Precisely. And this difference stems from those subtle variations in their structure. Enzymes, those highly specific catalysts, are key players here.
Right.
They can distinguish between these isomers, directing them down different metabolic pathways.
It's amazing how those seemingly small structural differences have such significant consequences on a physiological level.
Absolutely. Now let's shift gears and talk about D and L isomers in a medical context. Remember how we said most naturally occurring sugars are D isomers?
Yes.
Well, this preference has major implications for drug design and pharmacology.
Oh, right. I remember hearing about this in pharmacology lectures. It has something to do with how drugs interact with targets in the body, right?
You're spot on. Many drugs are chiral molecules, meaning they exist as D and L isomers.
Okay.
And just like enzymes are picky about which sugar isomers they interact with, drug targets, often proteins, can be very selective about which isomer of a drug they bind to.
So is it possible that one isomer of a drug might be effective while its mirror image is completely useless or even harmful? That seems like a huge deal for pharmaceutical companies.
It is a huge deal. It's not uncommon for one isomer to have the desired therapeutic effect, while the other might be inactive or even cause unwanted side effects. That's why the pharmaceutical industry invests heavily in separating and purifying the desired isomer of a drug. It's all about maximizing efficacy and minimizing potential harm.
Wow, that's incredible. It's a real-world example of how chirality and isomerism are not just abstract concepts, but have tangible consequences for human health.
Absolutely. Now let's move on to a more sensory example.
Okay.
Smell.
Smell. Okay.
Have you ever noticed how oranges and lemons, both citrus fruits, have very distinct smells?
Oh, absolutely. Oranges have that sweet, sunny scent, while lemons are more tart and zesty. But what does that have to do with isomerism?
It turns out that the molecule responsible for that characteristic citrusy smell, limone, exists as two enantiomers, D-limone and L-limone.
Wait, so the smell of an orange versus a lemon is determined by the chirality of a molecule.
It is.
That's fascinating.
D-limone, the right-handed isomer, is primarily responsible for the orange scent, while L-limone, the left-handed version, gives lemons their characteristic aroma. It's a striking example of how even our sense of smell is exquisitely sensitive to the subtle structural differences between isomers.
That's mind-blowing. It's like nature's playing a game of molecular aromatherapy.
I love that description. And this phenomenon isn't limited to citrus fruits. Many flavors and fragrances are due to chiral molecules. For example, carvone, another chiral molecule, exists as two enantiomers. One smells like spearmint, the other like caraway seeds.
Okay, I'm officially convinced that isomerism is everywhere, influencing everything from our metabolism to our senses.
It truly is. Now let's dive into a biological example of epimerism that's relevant to human health. Remember how we discussed glucose, galactose, and mannose being epimers of each other?
Yes, differing at just one chiral carbon. But how does that come into play in our bodies?
Well, consider lactose, the sugar found in milk. It's a disaccharide, meaning it's made up of two sugar units linked together, glucose and galactose.
Okay, I remember that from my introductory biochemistry class. But
Yeah.
what does this have to do with epimerism?
The key lies in the enzyme lactase, which is responsible for breaking down lactose into its component sugars so our bodies can absorb them.
Okay.
Lactase is incredibly specific for the galactose portion of lactose. It needs that galactose to be in the right configuration to break the bond.
So if the galactose were replaced with its epimer, mannose, would lactase still be able to break down the molecule?
Nope. Even though galactose and mannose differ at only one chiral carbon, that's enough to throw lactase off. It wouldn't recognize the molecule, and lactose wouldn't be digested properly.
Ah, so that's why some people have trouble digesting lactose. They lack the enzyme lactase that can recognize and break down that specific glucose-galactose linkage.
Exactly. It's a classic example of how epimerism and enzyme specificity are intertwined. It's also why those with lactose intolerance often need to take lactase supplements to help them digest dairy products.
This is fascinating stuff. I'm starting to see how understanding these subtle structural differences between isomers is crucial for understanding not only carbohydrate chemistry, but also human health and nutrition.
You're getting it. And we haven't even touched on enantiomerism yet, which plays a huge role in the structure and function of complex carbohydrates like starch and cellulose.
I'm ready for more. Bring on the enantiomerism examples.
All right, let's explore enantiomerism
Okay.
in the context of some really important carbohydrates.
Okay.
I'm talking about starch and cellulose, two polymers of glucose that play vital roles in the natural world.
Okay, starch and cellulose.
Yes.
I remember those from biology class.
Right.
Starch is the energy storage molecule in plants, right?
Exactly.
And cellulose is what gives plants their structural strength.
Yes.
But how does enantiomerism fit into this picture?
That's the key to understanding their different properties.
Okay.
Starch is made up primarily of alpha glucose units linked together.
Okay.
Remember, in an alpha enantiomer, that hydroxyl group on the anomeric carbon points downwards.
Right.
This configuration allows the glucose units to form a helical structure, which is great for compacting energy storage.
So it's like those spiral-bound notebooks.
Yes.
The alpha linkages allow the glucose chain to coil up neatly.
Exactly.
But what about cellulose?
Yeah.
I know it forms those tough, fibrous structures.
Right.
How does the anomeric configuration contribute to that?
Cellulose, on the other hand, is composed of beta-glucose units.
Okay.
Now that hydroxyl group on the anomeric carbon is pointing upwards. This seemingly minor difference in orientation has a major impact on how the glucose chains interact with each other.
So instead of forming a helix, the beta linkages create a different kind of structure.
Exactly. The beta linkages allow the glucose chains to form long, straight chains that can pack tightly together, forming strong hydrogen bonds between adjacent chains. It's like weaving those chains into a sturdy fabric, giving plants their incredible structural strength.
Ah, so that's why we can't digest cellulose.
Right.
Our digestive enzymes can break down those alpha linkages in starch, but they can't handle the beta linkages in cellulose.
You got it. We lack the enzymes necessary to break those beta linkages, which is why cellulose passes through our digestive system as fiber. But for herbivores, like cows and termites,
Right.
they have symbiotic bacteria in their guts that produce the enzymes needed to break down cellulose, allowing them to access the energy stored in those glucose units.
Wow, that's amazing. It all comes down to the orientation of that one hydroxyl group on the anomeric carbon.
Yes.
It's incredible how such a subtle structural difference can have such profound consequences in the natural world.
It really highlights the elegance and complexity of biochemistry, and it's a reminder that even the smallest details can have a huge impact on the properties and functions of biological molecules.
Well, I have to say, this deep dive into isomerism has been a real eye-opener. I feel like I've gained a whole new appreciation for the intricacies of carbohydrate structure and function.
Me too. It's one of those fundamental concepts that is essential for understanding so many aspects of biochemistry, from metabolism to cell signaling to the structural components of living organisms.
And I think our listeners will agree that it's a topic that's worth revisiting and exploring further. So before we wrap up, let's leave them with some food for thought.
I like that. Let's challenge them to think about isomerism in their everyday lives.
Okay.
All right, listeners, here's your assignment. Think about all the different carbohydrates you encounter in your daily life.
Okay.
It could be the sugars in your food, the starches in your bread, the fibers in your vegetables, or even the paper you write on. Pick one that you find particularly interesting.
Pick one.
Once you've chosen your carbohydrate, do a little research. See if you can identify any examples of isomerism at play. Does it exist as different anomers? Are there any epimers that influence its properties? Does its chirality play a role in its biological activity?
Those are good questions.
We encourage you to dig deep and see what you can discover. Share your findings with us on social media or leave a comment on our website. We'd love to hear what you uncover.
Yes, we would.
It's a great way to put your isomerism knowledge to the test
Right.
and see how this seemingly abstract concept has real-world relevance.
Well, on that note, we've reached the end of our deep dive into isomerism in carbohydrates. We've covered a lot of ground today, but we hope you've come away with a deeper understanding of this essential topic.
We've explored the 5 main types of isomerism: ketose-aldose, D and L, optical, epimerism, and enantiomerism. We've seen how these subtle structural variations can have a significant impact on a carbohydrate's properties and biological activity.
We've also looked at real-world examples of isomerism in action, from the different metabolic fates of glucose and fructose to the importance of chirality in drug design and the unique properties of starch and cellulose.
And of course, we challenged you, our listeners, to embark on your own isomerism adventures, exploring the diverse world of carbohydrates and the role that isomerism plays in their structure and function.
As always, we're grateful that you join us on this journey into the fascinating world of biochemistry. Keep those brains buzzing, and remember, the tiniest of structural differences can have the biggest of impacts.
Happy learning, everyone!
And now, to test your knowledge, here are 10 quick questions with answers and explanations.
Oh, fun.
What is the basic definition of isomerism? The answer is molecules with the same chemical formula but different structural arrangements. It's like building different structures with the same set of Lego bricks.
Good one.
What is the key difference between glucose and fructose in terms of isomerism? The answer is they exhibit ketose-aldose isomerism. Glucose has an aldehyde group while fructose has a ketone group. This difference in functional groups leads to distinct chemical behaviors and metabolic fates. What determines whether a sugar belongs to the D or the L series? The answer is the orientation of the hydroxyl group on the chiral carbon furthest from the carbonyl group. D sugars have the hydroxyl group on the right, while L sugars have it on the left. What is an enantiomer? The answer is non-superimposable mirror images of each other. Think of your left and right hands. They are mirror images but can't be perfectly superimposed. What is the difference between dextrorotatory and levorotatory isomers? The answer is they rotate plane polarized light in opposite directions. Dextrorotatory rotates light to the right, while levorotatory rotates it to the left. This property is used to identify and distinguish isomers. What is a racemic mixture? The answer is a mixture containing equal amounts of dextrorotatory and levorotatory isomers. The optical activity of each isomer cancels out in a racemic mixture. Define epimerism. The answer is isomers that differ in configuration at only one specific chiral carbon. Glucose, galactose, and mannose are classic examples of epimers. What is the anomeric carbon? The answer is the new chiral center formed when a sugar cyclises. It was the carbonyl carbon in the open chain form. What is the difference between alpha and beta anomers? The answer is the orientation of the hydroxyl group on the anomeric carbon relative to the ring. In alpha anomers, it's below the plane. In beta anomers, it's above. This small difference can impact the properties and biological activity of carbohydrates, such as in starch and cellulose. Last question, why is understanding isomerism crucial in biochemistry? The answer is isomerism impacts a carbohydrate's chemical properties, how it interacts with enzymes, and its overall biological function. Enzymes are often highly specific, recognizing and acting on only certain isomers, which has implications for metabolism, drug design, and many other biological processes.
And that's it for today.
We hope you enjoyed this deep dive.